experience in the classroom, and also publishing in teaching related materials. I'm going to tell you a little bit about my tale as a postdoc. And I went through a botany department where it was primarily older professors, the old professors who were extremely dedicated to research, but teaching as well, being outstanding teachers. And I did a lot of TAing as a grad student in genetics and botany, and I learned that I really enjoyed doing it. I came here as a postdoc, and one of the professors who was chair, who had hired me, and said, I was doing research, and he said, why don't you publish that as a teaching or to develop a lab exercise from that research? And that was the most novel thing anyone ever said to me. No one ever told me to even think about that, and I did, and it was one of my first teaching papers that was published. And that was really exciting to me because we were creating new knowledge in the fundamental research side of it, but we're applying it in the classroom to help students. And I often believe that that's more useful than some of the material that we published in terms of the impact. And that started my kind of career here in teaching. When I was hired as an assistant professor, jobs were frozen. I applied. I was applying for different postdocs elsewhere, and a position opened up here, and at the last minute, and I applied, and I got it. That was almost 30 years ago. The day after I was hired, I went over to see our dean then of teaching, and I said, we need to create a publication series that faculty who are devoting a lot of time putting together teaching materials can get some credit for it. Now, this wasn't, and I said, let's have a academic programs or a teaching publication series. It's not refereed, but it allows you to have materials, instructional materials that are presented in a printed format, and you get credit for what you do. It's useful for teaching workshops, labs, and we started that series back in, 1986, I think it was. And we published these. They printed these up, and the academic programs, I don't know if it's still used now, this is a long time ago, but this one was on shoot organogenesis, adventitious shoot formation, one on potato, forming microtubers in potatoes. You want to get students interested in something, show them a potato in a tube. You get them interested. I pass those out for a reason. Those are Venus flytraps, insectivorous, carnivorous plants. Oh, thank you. You want to get a student interested? Show them this. Oh, I'm sorry. Sorry, guys out there. Everyone see it out there? I will send you some. I'll get the address and I'll send you each a culture. I brought that because that's a endangered plant. It's a novelty plant. It's found in the Carolinas, North and South Carolina. And it's been collected in the wild. And it's an endangered plant. It grows in areas that are very nutrient poor. So it's evolved to be able to obtain an alternative nitrogen source by capturing insects. And there are different types of those plants, but I give you a copy of that. I give you a culture of that, and also a little handout that I give actually to grade students and teachers. So that's about a little below the level that we do in our labs. So, but it shows you the background of the culture and what to do with it when you take it home. As Monica mentioned, I have all these cool tools in the lab that I can use to hook students. I look for new ones all the time. And my time as a teacher here has been fun. I enjoy the classroom, enjoy interacting with people. I don't have 800 students. And I have about 25, especially when we teach class. So I'm able to do certain things with that number of students I certainly wouldn't be able to do with a much larger number of students. So I could have been a mechanic. I like using my hands. I learn by doing. I think, as Monica said, with labs, hands-on experience are critical to learning, particularly in these courses involving technology. And labs defined by a wide range of activities, not just what traditionally we think is in a laboratory, but it could be outdoor labs, it could be field trips, it could be internships, unique ones. So I want to talk about my class, how we do it, how we incorporate various aspects of learning in it that really makes it learner focused. We put the responsibility of learning on the students. We open up the lab to the students, give them I think a great opportunity to use the facilities to learn by doing. So I'm going to talk about that today and talk about some of my tips of how to make it work. Okay, Dr. Whiteside, you're correct. Okay, next. Okay, so the objectives are today, I want to talk about the strategies, as I mentioned, to enhance student learning in the classroom setting. This is with about 25 students. I want to talk about how you can strengthen your teaching portfolio as a postdoc. That's why you're here, too. You want to learn how to teach more effectively, but also you want to get the opportunity to be selected for that position in which you can teach, right? That's the reality. And hopefully you will enjoy it as much as I do in doing that. I want to talk a little bit about mentoring graduate students. How many of you work with graduate students now? How many of you who work with graduate students work with them with respect to teaching in the lab? Nobody. Okay, that's typical. There's lots of opportunity to get involved. And I want to talk about incorporating graduate students in the teaching, and as postdocs, you can do that. Now, it may depend upon your lab, the funding, who you're working with. You know, each lab is different. I'm always told the culture in each lab is different. So take what you can from this and see what works for you in your particular situation. And it's worked for me in the past and continues to. And the last thing I want to talk about, and Dr. Roberts, have you talked about this yet, or are you going to? Okay, good. I just want to mention from my standpoint about the scholarship of teaching and learning, and Dr. Roberts is going to cover that. Okay, next slide. As I mentioned, I started my career here as a postdoc, and a few years later, I'm still here, still enjoying it as much as I did. And I want to share those things I've done. Next slide. Let's define a laboratory. Monica talked about her lab, which is a tremendous enterprise with those hundreds of students. That's incredible. I got to come over and see that. I need to just come over and shadow you for a day. We can define it as a place providing opportunity for experimentation, observational practice, and a field of study. It doesn't have to be in a physical lab setting that we typically think of. Okay? Now, there's a wide range of disciplines represented here. So think about those situations, those labs that you may have an opportunity or make the opportunity to be involved in. Okay? Next slide. Brian Myers, I don't know, Brian, did he discuss anything? He had a good definition of a lab. An agri-science lab is a learning experience in which students interact with materials and models to observe and understand the nature of agriculture and its underlying biological, physical, and social science components. It's a wide range of experiences. You could be working on a computer. Anyone just work on computers here? Modeling? Oh, okay, we'll get modelers here. Okay, so that's a different situation. Next slide. I teach a combined graduate, undergraduate class. The expectations are different. Micropropagation of horticultural crops. And that culture that I gave you was produced through a technology called plant tissue culture. If you go to Lowe's, Home Depot, most of the house plants that you see there were produced in a commercial lab like this, AgriStarch, down in Apopka, Florida. They were produced using this technology. Micropropagation is a big industry in the United States. Globally, it's a big industry. And we have some very large labs in Florida, which we train students to work. We have students who worked in our lab that are R&D directors now for labs. But we teach the students the nuts and bolts, the technology for successfully, efficiently producing plants using micropropagation. And that culture tube that I gave you is an example. That's what they call a multiplication culture. There are many shoots in each one of those, probably 20 or so in each one. Okay, next slide. So in this course, it's a 4-credit class. We have 3 lecture periods in a 3-hour lab. And in the lecture, we provide the background information on the technology and then hands-on experiences in the lab. Okay. We try to, we don't try, but we do incorporate the scientific method in the lab. We also have self-directed projects that the students work on. They learn, they develop their toolbox of techniques, and then we set them loose in the lab to work on self-directed projects. And many of my colleagues think I'm nuts doing this, but we do it, and it works. It's a lot of work, but it's worth it. Okay, next slide. The process of micropropagation involves 5 steps, 5 stages, from having a plant you want to clone, you sterilize it. Micropropagation is defined as the rapid production of plants in a sterile environment on a controlled, defined culture medium. That gel that's in the tube contains all the components to induce rapid growth of the plants. Okay? And it's sterile, it's free of contaminating organisms. That gel is very rich, so the students have to learn sterile technique. We try to keep the bacteria out of the cultures here, not in the fungi, but we're not always successful. But the thing is, to complete the process, there are 5 stages. And the medium and conditions for each plant have to be optimized experimentally in each of the stages. It's establishment of the culture, multiplication, rooting, and then greenhouse, back into the greenhouse. If you have one plant, you get 10 new shoots every 4 weeks. At the end of 6 months, you have 1 million plants from that one. And that's easily attainable plant. So it's a production, it's a propagation. Next slide. So we have this lab that we've developed over the years, and our lab facility is great. We've been moving into the new building. We get the opportunity to renovate, and we have a really nice lab facility with transfer hoods, which work very well. But the lab that we've developed, we've refined over the years. We've made plenty of mistakes. We've refined it. And these are some of my hints. If you're teaching a lab class like this, these are some of the things that I want to share with you. Link the relevance of the lecture concepts to the lab activity. How many of you have ever had labs where you started the lab and you didn't have a clue of why you were doing the lab? Anyone have that? Thank you. In terms of understanding how it related to the concepts in the, let's say in the lecture or material that you read, it's critical the students understand why they're doing that lab. What are the objectives of that lab? How do those objectives relate to the concepts they've been introduced to, let's say in lecture or in their readings? It's critical that you keep that in mind when developing a lab. Develop effective and professional teaching materials. If labs are hastily prepared, the handouts are of poor quality, what does that do in terms of the student's perception? What do you think it does? Ah, okay, good. If the student, that's exactly right. The response was if materials aren't prepared correctly, the student perceives it's probably not of value or not worth it, and they're not going to be invested in the learning experience. And that's what I found. If you take the time to prepare well organized written materials, the students will recognize the fact that you're serious about it. And there's also another factor of credibility. If you project the image and hopefully you are prepared and knowledgeable of the topic, the students will recognize that and will become invested in themselves. If you're ill prepared, they sense that right away and then they don't become invested. I've seen that. So we do take time. I did not bring one, but I can bring one of our lab handouts. It's like a little paper. It has a background, introduction with literature, the materials and methods, all the graphs, data tables and everything, and follow-up questions and literature cited. So we try to keep it complete. Good question. How do we get students to buy in? I tell them in terms of when we start out that I will work as hard as I can. The deal is that you will work as hard as you can. We'll do what we need to do to make it work. Good example is when we do the labs. We just put strawberries out in the greenhouse for a lab for 1 week in January. We have to grow them in hanging baskets to do a shoot culture lab. And I explained to them the amount of time and effort and resources that go in to making this learning opportunity. And they usually understand that and appreciate that. But it's very easy, I see, for students not to focus, not to appreciate all the background and preparation that it takes to do what we do in the classroom. So, okay. My labs are like plays, acts in a play. Each lab is based upon a technique that they learned in the previous labs. It builds upon that. And it's an orally sequence through the labs based on that diagram that I showed you, that flow chart with the 5 stages. So they always know where they're at in the labs and why they're doing what they're doing. We always develop present demonstrations of how to do certain techniques before they actually do it. Then we reinforce it as they're doing it, and not just give them the handout and just let them try to do the lab. And then we offer learning opportunities that are more challenging, more and more difficult to do as they go through the semester. You don't want the students to feel they're not successful in the beginning. That's one of the little tricks that I try to do. It's not a trick, it's a strategy. You want to build their confidence. You want them to buy into this, that they can do it. They can do it. I teach workshops for teachers in the summer on introducing plant tissue culture in the classroom. And I'll ask the question, how many of you have done tissue culture in your classroom in high schools? And a bunch of them are raised. How many of them did it work? 1 or 2. And I'm never going to do that again. So we worked in trying to build confidence in our students and interest. Next slide. We allow our students, when they do a lab exercise, I'll say to them, what would you change? And they look at me and say, well, I'm just going to do the handout. I'm going to do A through Z and just complete the lab. I said, no, what would you change? Would you want to change a treatment, a procedure? And, some students don't want to do that. They want to do the lab. But many of them want, well, what if I did this? And I'll say, try it. And they get a little nervous and they say, well, that's not what you're asking me to do in the lab hand. So how am I going to be graded by that? I said, don't worry about that. And provide these opportunities for students to personalize it so that it really becomes not just a lab exercise and they're doing for Dr. Kane, but for themselves. They're on a journey of inquiry. Okay, next slide. Always provide the handouts before the lab so that they have an opportunity to review them. Some will have a quiz in the lab, which quizzes on the handout to make sure that the students have actually looked at that. And that's a pretty clever, very simple thing to use, because many times students will not look at the lab handout before. So I've done that. In preparing the lab, it's fairly intensive, not quite as intensive as 800 students, but make sure you have a checklist of everything that's prepared. We have grad students helping us. And then go through that and make sure that you have extra for slippage. You know what slippage is? If you just make enough for 25 students and you drop a rack, I dropped that rack of culture tubes, you're going to have short. And so always make some extra, because you never know. Review each step before you do it to ensure that all the materials for the exercise are available. Even having said that, I've had labs where, whoops, we've left a treatment out. And they just didn't catch it. And then always reserve a set for the instructor to demonstrate. These are simple things, but you'd be surprised how many people forget that. Next slide. Each lab handout is set up like almost like a paper manuscript. It has an introduction. It has materials and methods. It has the objectives, the learning objectives, materials and methods, literature, data sheets, includes everything. Now, I do that for a reason. In the class, we have the students get heavily into the literature in terms of reviewing papers critically. And this is one thing I see that students don't have a lot of experience with, in terms of they think anything that's published is good. And that's not, as we all know in here, that's not always the case. So I have them to critically analyze the papers, and I'll talk more about that in a minute. But I set up the lab handouts to look like papers, similar format. The only thing that's missing is the discussion and the results. So I keep that theme. It also includes a clear list of the materials. Many of our students have gone on to become teachers, and they go back and they do these lab exercises themselves. We have data tables, arrange time for post-lab requirements. Maybe they're coming in to collect data. You want to make sure that in there they need to know when they're collecting the data as appropriate. Have questions that they have to answer. Sometimes I have students collect these, then have references there. Next one. I think we skipped one. Okay. I mentioned the need to link what you do in the lecture, the concepts, to lab activities. And for years, for a while, I was trying to figure out how I do that in a neat package. And I said I'd never publish a book, and I never have, but I started publishing chapters in textbooks. I would have my colleagues be the editors of the books, and I would just write chapters. I started out with one chapter and two chapters, and I use this book now, Plant Tissue Culture and Development and Biotechnology. I have 5 chapters in there, concept chapters and also lab exercises. So it's nice for me to have those chapters in a book that we use, and then and students can relate to those concepts in those chapters to what we're doing in the lab. And I very closely have what the content in the lecture follow what we do in the lab. So they both support one another, okay? Next. I had mentioned about developing quality visuals, materials, handouts. We make graphs, didn't scan very well, but that we use, we do a synthetic seed lab, and we make plates that demonstrate the procedures so the students know very clearly. Okay, next one. I had said that the students are given the opportunity to change the lab, and it's had profound effects. We had one student, we were doing a regeneration in tissue from stem tissue, and the student looked at me and said, "Dr. what if I cut it down the center as a piece of a stem?" I said, "We've never done that." And then lay the half on the medium. She did that, and we got a 50-fold increase in shoot production. Unbelievable. Because the way that the tissue was cut made a profound effect. And it was just wonderful. So she did that and gave a presentation on it, and we incorporated it in the lab. Next. Okay. One of the things that you need to be aware of, and I know as postdocs you're aware that in the labs here, is safety. And this is particularly true with teaching a lab where you have students of various experience levels, attention, challenges, who could be in a situation to endanger themselves. And you want to make sure that you provide adequate safety instructions to the students. You want to promote safety habits. I have 4 grad students, and we're always reviewing the procedures in terms of the lab and re-reviewing them as we forget sometimes. You want to teach safety practices for each area of the lab, and I'll show you a slide in a second. Expect the unexpected. But one time, not with students, but with a grad student, we had a hood catch on fire. Because years ago, we used to use alcohol to sterilize the tools. We don't do that anymore. So they stood there and were wondering whether they should use the fire extinguisher. There's a HEPA filter in these hoods for about $1,000. So they thought the fire extinguisher would ruin the hood. Well, what I didn't tell them, I was in the library at the time, was it was a carbon dioxide fire extinguisher. You could use it, and they finally did use it. But it made me realize never leave anything unsaid in terms of safety, because the unexpected will happen. We use electric glass bead sterilizers now. We don't use alcohol. But the unexpected will happen. Next slide. So we use transfer hoods. It's our big equipment. These are just fancy air filters. And they sterilize their tools in this glass bead sterilizer. So that looks pretty safe, right? I don't see any place there where anyone's going to be injured. So from a safety standpoint, it looks good. So next slide. So that's our hood. Next one. Sterilize about 260 degrees. Pretty hot. We have these tools that are being sterilized. Next slide. So the hot tools, so burning is a threat. Next slide. Scalpels. And this is the one that we're always worried about. We make sure students wear pants. No open-toed shoes. If they drop it, hopefully it'll be deflected and won't go into their leg. We've never had a problem, but we could have a problem there. So these are our main things in our lab. Next slide. We use bleach, which could get in the eyes. Want to let students know where the first aid kits are, the fire extinguishers, and the chemical baths. These are things you may not think about. You may say, well, Kane, why is he reminding me about these things? But if something arises and the student doesn't know where to go or how to take action, it could be very serious. And if you're guiding grad students, they need to know. Next slide. We begin the lab period by providing an overview of the lab, and I go over with my grad students, because I work in the lab as well too, but many times I'll have the grad student manage the lab, and I'll be his or her assistant. And I try to instill professionalism in the TAs, because I tell them is these students are going to be looking at you, and they're going to be gauging you in terms of your credibility based on how you act, how you prepare. So our students always prepare before they go in there, and usually do a good job. Next slide. For about 5 years, 6 years now, we've been doing a little organized blip in the beginning, an overview of the objectives, the procedures. We have a 3-hour lab, but we don't go an hour and a half. We go about 12 minutes. And we'll put that up. I have an LCD projector in there. You have the hoods. And we've got it set up pretty nice so the students can turn and look and see the procedures or how to make the cuts. So we do a lot of things that are visual. Okay, next slide. And that overview will provide the objectives of why they're in the lab for that unit and what we are doing during that lab period. They'll always have that to see up there, and also the procedures. Next. One of the things that we emphasize over and over again in a lab of this nature is the scientific method. One thing I found is that students don't have a lot of experience with respect to the scientific method. Now, I've had students, I shouldn't say it with the dean here, ask me what a mean is. Have you had that too? Yeah, okay, good. Wow. And so we work very hard to have our students, both undergrad and grads, understand the scientific method, the literature, how to search the literature, how to evaluate the literature critically, how to collect data, design experiments, collect the data and analyze it and interpret it. So we work very closely in these labs, that each lab exercise, when written up, is written up like a scientific paper. Next. When you do the demonstrations, this is where sometimes we provide variations that could be done. You may change something. One of the biggest challenges we have sometimes in doing biology labs is having appropriate plant materials, or it could be animal materials, for the lab. Sometimes that's not really certain material may not be available, so you have to change it. So if you're modifying procedure, so you want to make sure you do that during the demonstration, the overview. And again, any particular safety issues for that lab, bring those up. Okay, you want to tell the students how to get started. Never provide them with a handout. Say, go for it. Step 1 through 8, complete step 1 through 8 or something. Really provide them some background to get started. Okay, next slide. While they're doing the lab exercises, we're very interactive. I don't like standing up like this. I'd be over in here with you guys if you were doing the lab exercise. But you want to supervise them, you want to encourage them. You may help them while they're doing the procedure. You may demonstrate it. You may isolate a shoot tip, let's say, for them and show them how to do it. Each of them will develop their own technique, and you never want to take that away from them, because they all have different preferences of styles of learning and doing procedures. And you can also suggest an alternative method. Next slide. We have them work in groups and pairs, depending on the lab exercises. That works fairly well for doing experiments. We have multiple treatments. It's a much more efficient way to run those lab exercises. And then they prepare questions and share their responses and work in groups. That's always an interesting learning experience for the students in this lab.
Could you rotate?
Yes. Yes. One of the pros is sometimes when you have two students, one may have experience and will dominate the other one so that they want to get the lab done, so they can kind of take the number one role. And in rotating them, that helps eliminate that. I haven't really seen too many negative experience with it. It's different each time.
Everything is done.
This is in the lab, yes, during the lab period, yes. Okay, next one. Some students need extra time, they're slower. At the end, we always reflect in terms of take 10 minutes to talk about what they did, why they did it, what the results were, and what the students' impressions were of the lab experience, what they would do different. We talk about it, that's a good question. I walk around with a wireless mic. It's a lot of noise in this with all the hoods, and we talk about it as they're working. Most of the students, there'll be a few, if I have 14 in one lab section, there'll be 2 or 3 that are still working, but they'll listen, and we'll talk, and they'll respond while they're working. But we'll do it without, if we can stop and sit around, that we have a central table, we will. But if not, we'll do it as those students are finishing up. That's a good question, though. Then we have them clean up. That has to do with responsibility. I'm one of these old guys. I mean, and if they make a mess, they clean it up. And sometimes, most students is great. Some students really don't adhere to it, but they have to clean up the hoods and everything. Okay. Next one. They collect the data. They analyze it statistically. Anyone use JUMP in here? JUMP's a good little, for SAS, it's a very simple program, and we have our undergraduates. It creates a lot of stress, I see, with undergrads, because a lot of them have done it, but we show them they can do it, and it's on, approach it, show them how to set it up and analyze it, and it works. They say, hey, I can do this. So they analyze it, they interpret the data, and they discuss it. Next slide. Okay. How are we doing on time here? There's 5 steps in the micropropagation process. The students have learned various nuts and bolts of achieving each of these steps, okay? By lab exercises. By about the 5th lab or so, by then I've had that they've selected a plant of their own choosing, which they're going to use this toolbox that we've created, these techniques, for them to successfully establish their own cultures and work through the process, okay? This is required. Students have to do this. Now in lecture, the first week they pick the crop. Let's say it's strawberry, okay? A Venus flytrap. They picked a crop. They're going to do a literature review. They'll write a literature review, a 10-page lit review, which will incorporate at least 10 recent papers. They will critically analyze the results of those papers based upon each of these stages. Do they contain all the information? Are they good papers? Are they quality papers? So they'll write their lit review. And then at the end, based on that lit review, there will be a 1-page protocol based on the literature that they would follow, produce that strawberry crop. They're going to pick the culture media that would be used in each of these stages. And then what we do is we let them go in the lab on their own time and actually establish their own plants. Now they get so many points to their final grade if they achieve each of these. Up to 4 points to their final grade. If they go back into the greenhouse, they get 4 points to their final grade, which is a lot of points. We've had students every semester who are able to do that. We provide the hoods on the days we're not teaching the lab. We have a sign-up sheet. We have media. We make up a very different types of media. We can't let them just go in and make up their own media. This doesn't work in the lab. But they come in on their own time, and they establish the plants that, the crop that they picked. And then they write up a report at the end. I've had students who have written publication quality in one semester reports. That were, one did one on mint that was unbelievable. He did so many experiments. The plant grew fast, but the student, he was very quiet, he was extroverted, and he just blossomed. I've never seen anything quite like that today. In fact, he talked at the Teaching Enhancement Symposium 3 years ago about the self-directed project experience, and he was just, I've never seen anything quite like that, just blossomed. So it works because students personalize it. Okay, next slide. So they go through the procedures. I forgot I had, this is strawberry, in fact. They go from putting it in culture and going into the greenhouse. I just show you an example there, okay. Next slide. So this is basically what I just mentioned about the procedure for the self-directed project. They do the review paper. They come up with a proposal of a protocol they're going to follow. Then they go in the lab and they do it. Sound familiar? Sound familiar, right? Aren't you doing that now? And then they write up the final paper. Now they've done lab reports previous to this, written in the format of a published paper, so they know how to do it. It's a lot of work, it is. And I've never had a student complain that it was a lot of work, but I recognize it is they do spend a lot of time. Next slide. Florida is unique because we have quite a few labs, commercial labs in Florida. Down in Apopka, AgriStarts, there's 3 of them. There's a huge one out in the Panhandle, Oglesby. There's some others in the Apopka area. So we're able to go to those labs, and the students see it in action in the real world, and from the commercial standpoint. They see it. And that's very, very important, that they be able to tie the real world application to what they're doing in the lab. And we many times will go to 2 labs during the semester field trip. So we integrate that as well. Okay. Next one. This course is very expensive. Scalpel blades are about $35 a box. We go through lots of those. The student fees, lab fees, cover quite a bit of it. We have the equipment fee, which helps cover, replacement costs for repair of the autoclave and things like that. So that helps tremendously. But also we get donations. Next slide here. From different companies. And I'm not bashful to ask. And they usually do help us because we're training their students. We have many of these. Oglesby, AgriStarts, there are people from our program that are employed there. So to their advantage too. And it works quite well. Okay, next slide. All right, let's take a couple minutes, and I want to talk about working with grad students. And some of you work with grad students. We were all grad students. And I want to ask a question, pose the question, if you were teaching a class, or you may be teaching a class, what do you think considered the primary role of grad student in the class? Let's say you're a professor. You have the job, the dream job, the tenure track position with a teaching component. You have a bunch of grad students, you're teaching some classes. What do you consider the responsibility of a grad student? Or it could be an undergraduate. We have undergraduates who are TAs too, but teaching assistant in your class. What is your responsibility? with respect to what their involvement is. What do you, is it just labor? You ever feel like it was just labor? I don't know. Excellent, yes. A role model. A grad student can be a role model. So the way that they perform is critical. It's been done to Excellent. That's right. You provide them to be creative in terms of the way that they present the information. They know what they need to present, but if you allow them the flexibility to be creative, develop their own techniques, that I think that is a good. That's excellent. Yeah, no, they're the intermediate level. They're more closer. They can relate to them closer than the professor. That's very, very important. Should you provide opportunities for the graduate students to develop in the context of teaching the labs or maybe even lectures? So you can provide a little bit more added attention and help them and mentor them. Okay. What about you as postdocs? Uh, the program manager or director, and I know it's different in each lab, but those who employ you, what are their feelings in terms of our philosophy? You're here. You're spending 2 hours a week learning about teaching, right? Yes. It's all the same. Same here with us. Right. Mm. Oh wow. Great. Excellent. We already answered some of these. You had some good suggestions how you would guide students. Any other ones? I encourage my grad students to be actively involved. They're on committees in the college. They're on the curriculum committee. Two of them were on the curriculum committee. They're involved in various committees. I encourage them to be very, very active in teaching. They come to the enhancement symposium and they get involved in professional societies. I'm a member of the Society for In Vitro Biology, been a member for 30 years, and our grad students get involved with the teaching aspects there and also graduate professional development. We ran a workshop on teaching effective plant tissue culture labs, and they were the conveners. We had it was a 3-hour session, and they convened it. It was an international meeting, so I try to get them involved in all aspects of in different levels of teaching, and that helps them in terms of when they go into the classroom, and also make opportunities for them to teach lectures as well. every opportunity I get. And what they also do is they've taken the classes over in Ag Ed, which has been very, very helpful because we are not trained as professional teachers.
Not all.
Yes.
Obviously.
Some don't. That's a good point. A lot of them do for some reason. I don't know. It's just, but and that's fine. And I work with them in different ways than that. And it's very, very important that you understand what the aspirations are of the student because they're different. They're different. I have one that wanted to do outreach in terms of working with in recruiting, who's now at a college now recruiting. He was a master's student and he didn't want to do research. He found that out, but did a good job as a master's student. And he didn't want to teach in the classroom as much as he thought he did. That may change. He's young, but now he's working as a recruiter, as JJ. But you got to respect the students. It's not doesn't fit all. One mold doesn't fit everybody. Yes. Good point. That's a good point. Okay, next. We've been talking about mentoring, and that's what we've been talking about is mentoring students. And as a postdoc, I mentored a couple of grad students. When I came here, the professor who ran the lab became the chair. Didn't have time to run the lab, so I kind of had a quasi-faculty role and guided the grad students, so I learned a lot. Because when I came to the University of Florida, I wouldn't be here. I wouldn't be here in academia if it wasn't for two professors who, many years ago, took the time to help a city kid learn the value of education, developing confidence in themselves, and I have two photographs on the wall. One just passed away last year at age 98, Dr. Caroselli, and the other one was an advisor of mine for my master's, and they made a tremendous difference. So mentoring is very, very important. We have a powerful role here and responsibility because we have a tremendous effect on people that lasts for a long, long time. So being a good mentor, whether it be grad students, undergraduates, is very, very key. Next slide. There's a book online. I have a copy if you want to. That is pretty good about mentoring. It's National Academy of Press. It was free for a while. I have a PDF of it. If someone wants to. I don't know what the legalities are of sharing that, but send me your email, and I can provide you. But it's a good kind of little light overview of mentoring. Next slide. All right, I'm going to end here. And if anything, I think teaching is extremely important. I think but why can't you publish in the area of teaching? This is something I learned early on. I'm glad I did, because when we think of research, we think usually of separating teaching and what we traditionally call research as being separate, and they're really not. Oh, thanks. We're in an R1 university. We hear a lot about research, right? This university is moving towards preeminence. I'm not sure what that means, personally. What role will teaching, quality teaching, play in that as we move forward? That's the questions that are being asked now. You'll take a faculty position, it's tenure track, and you're going to be assessed. I'm 70% research, 30% teaching. You surprised by that? I'm mostly research. My problem is I never look at numbers. That takes it fun. I used to say I'm 100% research and 100% teaching. When we used to, deans just come over and they just look at me and then they smile. They knew what I was meaning. But I enjoy what I do. I integrate it all. And I've done that since I've been here. And you can publish your teaching related efforts. You can share it. You know, the old way was, you know, you go in the classroom and you teach and that was your thing. You never shared it. Just with the students. Certainly no one else. You didn't want anybody in your classroom looking at what you were doing. You know, we do that. We go in and we assess each other. We look at and share how we teach. And things have changed. We are sharing what we do. We're doing that right here. And I want to share with you one, for me, was a very, very important thing. And next slide. And that is the Scholarship of Teaching and Learning, SOTL. And Grady Roberts is going to talk more about this. And I guess you're going to do the same thing as you did before, maybe? If anything, I would listen to what he's going to talk about. The Scholarship of Teaching and Learning, and I won't go into the background because he will, is the systematic study of teaching and learning, public sharing and review of such works through presentations of publishing. Publishing your teaching, I'll just put it simply, publishing your teaching related material. Could be surveys, could be lab exercises, that's what I do. Could be studies, experimental studies, you've done the teaching, learning, assessment, wide range of topics, publish them. Think about that when you're teaching, applying and sharing through publication. There are various examples of this. Next slide. You're sharing and contributing to this ever developing knowledge base with respect to teaching and learning. You're probably looking at me and say, give me an example. I'll give you a couple examples of what we've been doing. And teaching today is viewed as a very serious intellectual activity. And I guess we're having conversations of what scholarship is on this campus. But it is a very, very important component of your position if you're in a university, and you should take advantage of that. And next slide. We're running out of time, so I'll just ask you, has any of you published anything that's teaching related? Not yet, okay. Have you thought about that? We got a yes here and no movement anywhere else. How about out there in the state? Anyone publish anything? No?
No.
Has anyone talked about this? No.
No.
Sure.
So when you publish it, how is it? I guess different
Yeah, well, okay. Yeah, I've not published a book. I published book chapters, and someone else could probably address. I publish referee papers, and it's whatever it takes for a regular referee paper. Yeah, it's not that long. Books, I don't, and it depends on the publisher, but I've contributed to books, and I'll mention, okay. Next slide. So we don't have too many examples here. People have done this. So what has Kane done? I've published book chapters, lots of book chapters. I always said I'd never publish a book, and I haven't, and I won't. I did have a chance to do one. I decided not to redo a book. But I've published many book chapters, which include concept chapters. which include lab exercises that we share with others. Anyone here at Carolina Biological? They sell kits, very expensive kits. And I've given them my lab exercises over the years, and I probably couldn't do this now at the university. We have to write up an agreement, but I've given them. They've sold many of my kits all over the world. I've never made a penny on it, so I just gave it to them because if we share it, that's good enough for me. My wife always says, oh, we should have got something for that. But the point is, this is one of the kits that we had put together. We had published a paper on it, and then they sold the kits. So we do that. We share that way. We share it in book chapters. Next slide. This is a lab exercise on micropropagation of roses. Everybody knows roses in here, right? Did you know roses will flower in the tube? You want to get a student interested? Show them that. It's almost as good as the Venus flytrap. They're riveted by that. So I have all these little hooks that I hook these students in and suck them in and get them interested. But this is a book chapter. It counts. Does it count as much as a referee? Probably not. But I'm willing to bet that this chapter has more impact on more people than many referee papers would be. Okay, next one. I already talked about that series that we had in the college some years ago. And these are non-refereed. Another one that you have as a PDF is a lab exercise on orchids that we published in the plant cell tissue organ culture in 2008, I think it was, 2008. This is the first refereed teaching paper in that international journal, the first one. They published it. It's in color, and you have a copy of this. So this gives you an example. That's a refereed publication on teaching. And there are many journals. I think you mentioned some of these journals that, depending on your area, will publish teaching-related material, refereed material. So take advantage of this. Share the wealth. Okay, next slide. There are two PDFs that everyone has on a little bit different. One was with Sandy Wilson and Max Thedford, where they did provided a publication on new strategies for teaching plant propagation by distance education. So if you have a new idea, a new technique, publish it. Don't rule out anything. Share the information. It may be very valuable. There's another one, Dr. Roberts was on with Tracy Arani, which is evaluation of the effects of multi-site distance ed of knowledge gained in plant propagation course. This was a one related to this. So there's lots of opportunities. Look at that. Look at examples of what you can publish. Okay, next one. Okay, next, I think we're about over. The key is to collaborate. Collaborate with other instructors. We do that in our department. We publish papers together. And I think I'll leave you with this. I think the best weapon when you get your job, or you go back into your lab now. How many of you have a lot of grad students in your lab? Not too many, okay. All right, well, when you get your own position, you have grad students, this is the best weapon, okay? Are the grad students. It's fun, it makes you productive, it makes you good, and it provides you an opportunity to work with them and develop a productive program with a little bit less effort. And that's by working smart. So I hope I've given you some ideas. If anyone wants to come over to the lab, just send me an email, and it's right there. and I'll show you the lab, and I'll share anything I have, teaching-wise or what have you. But I'd love to you come over and show you what we do. It's Micropro, Micropro at ufl.edu. Micropropagation, that's what that came from.
Monica, I.
And I would contact Monica, but she certainly has the experience with very effectively teaching extremely large classes. Well, I've enjoyed coming over here. I wish everybody the best in terms of landing that perfect position or another postdoc. And if I can in any way help you, just give me a call, and good luck with the Venus flytraps. And the guys out in the state, I will get the address and I'll send you one. Any other questions? Sure. I would, they're starting to form roots now. I would let them go about 3 more weeks, and then you can. Sure.
As we kind of draw closure today, I want you to think back, and so the lab work that and working with TAs and GAs Monica does is on a different size and different scale than the work that Mike does. But I think at least I heard some common things that they said that might be helpful for you guys. And so thinking about the two presentations, what are some common things that you guys heard from Mike and Monica?
Oh.
Okay, so what are some things that you guys, okay, what are some things that you guys heard Mike and Monica say that you think, when I get my lab, my teaching lab, this is what I'm going to do? Organized and prepared at all times. Okay, go ahead at a distance. Go ahead. Okay, so maybe some other examples in here. Two hours listening to experts, and you guys don't have to take away message. Yeah, I mean the stuff that you guys can do in a lab, it's for most people it's really exciting. You guys are in your profession, but you want to be there. Obviously, something puts you in there. So think about how you can put your students, particularly if you're teaching like introductory labs, where they're not upper level students in your field. They're students that are just kind of exploring the science in general. Yep. Yeah, that's a great observation. It does take work to put into it, but if you put the work into it, you'll get it out. And Mike, I do encourage you guys to go visit Mike if you get the opportunity. 70% research, 30%. There's a lot of people on this campus without appointment think teaching is a nuisance and try to get out of it. Mike doesn't. Mike built it in. Go ahead. Yep, excellent. And I heard a couple things that a couple notes I jotted down is that in both the way they structure their labs, there's an opportunity for the students to personalize it. So it's not all just cookbook experiments. At some point in the labs, they have the opportunity to make it their own. And that they both really emphasize the real world application of these skills that you're learning. How can you take it Any final comments? Yeah, and we do have a couple of folks too, and Mike does it to some extent, but where we have faculty that are integrating their students in a real way into their research program. The students are out collecting data that can be meaningful to your own research. Monica mentioned that. 800 students? Can you imagine if you could get 800 people collecting data to support your program? That's a pretty, as long as you train them in a way that they're collecting good data, that's a pretty big way to make an impact on your program. Okay, so a reminder, logistics-wise, next week, unless you hear different, we're going to be in the microbiology building. Also, you've got some resources online to start working on your teaching philosophy statements, and we're happy to provide feedback along the way if you like. And but you do have a draft of a teaching philosophy up sometime in the next week. So next week, a little bit of motivation. You have a draft of your teaching philosophy due next week. All right, so thank you, everybody, and we will see you all next week.
Okay, so that worked out better the second half?
Yeah, it was much better.
Okay, good. So depending on who's presenting, we'll get more interaction, I think.
Yeah, that's true.
Okay, all right. Well, I'm not here next week. I'm at a leadership training, but Natalie, let's keep our fingers crossed. The new building and everything, I'll try to get some tech support behind her as well. So, the
Okay.
website working out for you guys as well? You see the materials we put up there?
Yes.
Okay. All right. Have a good week, and we will. I won't see you next week, but the week after.
Okay. See you.
All right. Bye bye.